Good morning, everyone. Welcome to Asthma Going Rounds. Today, we're going to hear from Ayobami Akenroye, who's in the Division of Allergy and Clinical Immunology. And she's going to be talking about predicting clinical responses to asthma biologics. This is live webcasting. You can submit a question to our speaker by entering it into the chat. CME credits now being awarded by the Mass General Brigham Office of Continuing Professional Development. You can write to MGB, you can go into MGB, PCD, CPD rather, at mgb.org. CPD stands for Continuing Professional Development. So MGB, Continuing Professional Development at mgb.org. To set up texting for your attendance, you can go to https://cpd.partners.org and you can log in. If you're a Mass General employee, you can choose Mass General employee, you can log in, or you can choose visitor login if you don't have an MGB username and password. There will be a bunch of fields to fill in, and then you can click My Account, edit the profile and your professional type, and give them your mobile number, and you can click Save. And once you've done that and you're registered, you can save that text number to your phone. Add the phone number 857-214-2277, which will be in the instructions to the contacts in your phone. And that's the number you'll use to get CME credit in the future for any of the MGB things. So the talk today is predicting clinical responses to asthma biologics. The learning objectives are identify factors that influence patient responses to the monoclonal antibodies that are used to treat asthma and making rational choices deciding to initiate treatment of asthma biologics. Again, the SMS code for attendance today is NOYSEY. So it's that phone number, NOYSEY. Chris Fant and myself are the organizers of this set series. These are my financial relationships, and Chris doesn't have any. And Dr. Akenroye hasn't reported any financial relationships. If there are any technical problems, you can send an email to BWH webcast troubleshooting at partners.org. So BWH webcast troubleshooting at partners.org. So it's really a personal pleasure to introduce Dr. Ayobami Akenroye, who's a member of the Division of Allergy and Immunology. Ayobami is a physician scientist in the Division of Allergy and Immunology, and she has an appointment at the Channing Division of Medicine. And she's a PI of the Drug Use Lab, focused on biologic utilization, safety, and effectiveness. Ayobami, we're very much looking forward to your talk about predicting clinical responses to asthma biologics.
Thank you. Thank you, Dr. Israel, for that introduction. Thanks, everyone, for joining us. So if you do see me, if it seems like I'm not looking at the screen, it's because I'm looking at another screen that has my presenter notes. I couldn't figure out how to do it here. So it's a pleasure to be here again. Thank you for joining. These are the objectives. I will briefly review biologic therapy in asthma, highlight the currently approved biomarkers, and I'll spend the rest of the time reviewing emerging evidence on other biomarkers and future direction in biomarker research. So we know that asthma is a heterogeneous inflammatory disease of the airways, and that in itself presents a challenge, because what that means is that it differs from patient to patient. In this figure, would see, which has been cited a lot of times, we see that now often we talk about asthma as Th2 high or Th2 or T2 high or non-Th2 asthma or T2 low asthma. However, there's a lot of overlap in this phenotypes under each of these endotypes. So for instance, we'd think about obesity-associated asthma being non-Th2. However, there are many patients with allergic asthma or eosinophilic asthma who are obese. And so this classification, as much as it has helped us a lot in understanding mechanisms in asthma, they're not perfect. This categorization isn't perfect. We do know the multiple pathways are dysregulated in asthma, and these include Th1 or Th2 pathways. What you'd see for in this figure is that there are many cells, many cellular players in asthma, in underlying asthma. We have Th2 cells, we have B cells, we have mast cells, and commonly in severe asthma circles, we talk a lot about the eosinophils. Fortunately, we now have various therapies that target these pathways. So most of these target either a cytokine or a receptor. So one of it, the one that works furthest upstream, is tezepelumab. It prevents TSLP from binding to its receptor, and by working further upstream, is effective in both type 2 high asthma, which is to my left, and also in type 2 low asthma, even though most of its effect is in type 2 high asthma. We have dupilumab, which in addition to tezepelumab, some of us might consider it a broader spectrum respiratory biologic because it blocks IL-4 receptor alpha, and in so doing, it blocks IL-4 and IL-13, which are two of the canonical T2 cytokines, and is a potent blocker of T2 inflammation and effective in a lot of T2 diseases, including asthma. And then we now have 4 biologics in the anti-IL-5 or anti-IL-5 receptor category. So interleukin-5 is a primary cytokine responsible for or active in eosinophil maturation, activation, differentiation. And in so doing, by blocking this cytokine, we can improve eosinophilic asthma. So as expected, these biologics are approved in eosinophilic asthma. And then we have what I consider the grandparent of all respiratory biologics, and that's omalizumab. That's an anti-IgE therapy. And this was approved over 20 years ago. It blocks IgE prevents IgE from binding to the high affinity IgE receptor, and it's effective in allergic asthma. What we do know is that several of these biologics can be reasonable choices for the same patient. And this is where biologic research or biomarker research becomes important. So in clinical trials, these biologics performed really well in improving asthma-related exacerbations, improving lung function fairly to modestly well, depending on the biologic, improving asthma-related quality of life, and many of them are oral corticosteroid sparing. However, when we use eligibility alone and baseline biomarkers in saying who would be eligible for what, for instance, in this study that is now outdated because we have 2 more biologics on the market since this was done. But what we do see is that even then, there was a huge overlap in patients eligible for these therapies, where sometimes 70% overlap between the populations. And so a lot of time, biologic selection now is often probabilistic, not deterministic, where we think, okay, this patient, based on their biomarkers, they're probably likely to do well on this. We start the biologic, and sometimes they do well, and sometimes they do not. And most of these biologics are approved for the T2 high endotype. And just you can see here that, for instance, for patients who have allergic asthma, we have option of the anti-IgE, dupilumab, the anti-IL-4 receptor alpha, or anti-TSLP. For eosinophilic asthma, we have the anti-IL-5, anti-IL-4 receptor alpha, and really anti-TSLP also approved in that group, or quite effective in that group. But when we come to T2 low asthma, we really have just anti-TSLP. And as I'll show you briefly, the anti-TSLP is actually most effective in T2 high. So definitely the T2 low asthma category is an area of need. So you might ask, which biomarkers guide biologic selection currently? And from this flow chart from this nice paper, I would see that the eosinophil count and FeNO are the prime biomarkers that are being used now. So often you'd hear a threshold of 150 cells per microliter, even though we know that the further you get away from that, at least greater than 300 might be preferred for many of the anti-IL-5s and even the anti-IL-4 receptor alpha and anti-TSLP. And then we use this fractional exhaled nitric oxide, which correlates also with corticosteroid response, eosinophilic inflammation in the lungs, even though it's really associated with IL-13. And we do see that sometimes we use that also in guiding our selection of biologics. But what you will see down here is still the recommendations. There's still multiple biologics for a lot of the boxes. So there's much more work needed in terms of selecting these biologics, to guide selection of these biologics. But we do know that we can predict response at baseline somewhat So most of these biologics, they work better in patients with higher absolute peripheral blood eosinophil count. So the AEC, the peripheral blood eosinophil count, as we'll see here in this study, and this is for tezepelumab, you see almost this stepwise improvement in response as eosinophil count increases. We see similar things for pheno, even for tezepelumab, which again is approved for T2 low asthma as well. But you will see that the effect there is not as great as the effect in more T2I disease. And then when we look at dupilumab, anti-IL-4/13, and this is also similar for the anti-IL-5s, we do see again this stepwise improvement in response based on your eosinophil count being high. And so really the absolute eosinophil count predicts response to about 6 of the 7 currently approved biologics. So as you can see, that's not as helpful in guiding selection. And then we also know that pheno, the higher the pheno, the better the response to dupilumab, tezepelumab, and also, as I'll show briefly, partly to omalizumab. So in this study that pulled some of the randomized trials on dupilumab, if you look at on the y-axis, we have the exacerbation rate. On the x-axis, we have pheno. What you see here is that the separation between the placebo group and the dupilumab treated group gets wider as pheno goes higher. So we know that. But we also know that even though we often do not quote this, but really the anti-IgE is also more effective in patients who have higher pheno, as shown in this study here. Okay. So I think it's important to mention that for, IgE is a poor biomarker of response, even though people will often think that it should be a great biomarker for omalizumab responses. Omalizumab targets IgE. So IgE is important in telling us who can receive omalizumab because it's part of the eligibility criteria, but it does not consistently tell us who will benefit from it. So many studies have shown that higher IgE may associate with response, but those have been inconsistent. So the results are mixed, and IgE has not been established as a reliable predictor of response to omalizumab. And we think part of it is probably because your circulating IgE might reflect your sensitivity to things in the environment, even if it's not specific IgE, but not necessarily disease activity. And I think that oftentimes we miss that in predicting response. So I already mentioned that pheno is a predictor, but we do know across cutoffs that we might be more interested in. In adults, we usually consider the 25 to 50 range as intermediate range or indeterminate, and then less than 25 as the not, pheno is not elevated, the low range. But you see here that even if you use that cutoff in non-19, pheno is still associated with response to omalizumab. And your eosinophil count, which we often don't think about because omalizumab, again, is approved for allergic asthma. But at least in this cohort, it was also more effective in patients who had higher eosinophils, which here they used a cutoff of 260. However, defining response in itself is complex and multidimensional. So the domains that have been traditionally used in assessing response to these biologics in clinical trials and in observational studies have been exacerbations, which often includes whether you went to the ED or you were hospitalized or you was urgent care, you were discharged home. However, we do know that there are many other options that are important to patients, patient-reported outcomes, symptoms, like the asthma control test. We know that lung function is also important and can predict mortality, an independent predictor of mortality. And then there are other outcomes like oral corticosteroid use, which oral corticosteroid use, as we know, is associated with a lot of side effects and untoward effects. So even though we use exacerbations frequently, there are other outcomes that are of important, but response really and prediction of response is going to depend on whichever one we choose. And we know that, more and more we're getting to hear more and more about mucus plugging being another important outcome that maybe we didn't really think about before. So To underline this or underscore this point, that response is actually complex and multidimensional, and these response domains do not always align. And so the fact that a biologic did really well in improving lung function does not mean it would do as well in improving CACQ, at least from what the randomized control trials, which sort of sounds counterintuitive, but that's what the randomized control trials showed. And so in this study, we used Bayesian meta-analysis to pull estimates from the seminal trials used to approve these biologics. So we looked at the anti-IL-5s and the anti-IL-4/13 anti-TSLP. So biologics approved for eosinophilic asthma, and we limited to the subgroups with baseline eosinophils of 300 cells per microliter or more. What I want you to see, so these plots are showing the ranks, how well these improved exacerbations is what is on the y-axis, and then on the x is how well it improved this asthma control questionnaire, or how well it improved lung function on the C plot. What you see is that in terms of improving ACQ, mepolizumab across all the trials, and this has been replicated across various studies, others who have pulled these estimates either at the study level or even having access to individual patient level data, that mepolizumab led to the greatest improvement in ACQ. However, if you look to your right or to my right, you'd see that mepolizumab actually performed the worst in improving lung function. And this is, it's conceivable the biomarkers associated with one domain, say exacerbations, might not be the same biomarkers associated with another domain, say lung function. So prediction accuracy is going to be affected, again, by the response selected. And I think it's important as we go through the talk today to think what response, how did they define response, because that might have affected the results. And also might be affected by the omics domain. So is it metabolomics, is it proteomics, is it genomics, might have implications in the predictive accuracy of the model. And, something else not here is also the sample type can also impact that. Was this sputum, was it bronchoalveolar lavage, or was this PBMCs or blood? So often in many of these studies, response is commonly defined by the percent reduction in exacerbations. And you see that often we dichotomize, and people often use the threshold of 50% reduction. What you might think is, just what a lot of us also think is that is an imperfect way to define response, because response is actually, even exacerbations is on a continuous scale, it's a count measure. And so when we dichotomize, we do lose information, but often it's easier to say, responders had this elevated and non-responders. So that's another limitation to studies and something that we have to think about as we appraise the evidence. The patients probably are more on a continuum of non-response, partial response, or complete response, rather than just responder, non-responder. So that's like where we're at now, the validated biomarkers, FeNO, the peripheral blood eosinophil count, IgE not being as predictive, but now moving into areas that are still under investigation and that might help us in identifying other biomarkers. So I would start with genetics or genomics. We know that asthma has a strong heritable component, particularly childhood onset asthma. So prior studies have identified multiple genetic variants in key pathways associated with asthma that may influence asthma development or severity. And we can hypothesize that these may influence biologic response. But just the bottom line is that no genetic marker is currently validated for routine treatment selection. So in this GWAS, using the UK Biobank, the authors showed that genetic loci, multiple genetic loci associated with asthma, and we already know that. But what this paper added was that they looked at childhood onset, variants associated with childhood onset asthma, so less than 12 years, or adult onset, which they defined as older than 25 to 65 years of age. And they looked at the overlap between these variants and also what was unique about this variant. I've tried to annotate some of the things that might be of importance to us, like, ST2 region, which is the receptor for IL-33, one of the epithelial-derived cytokines or alarmins like TSLP, the TSLP region, IL-13. So you might be thinking, wow, this is great, because we do have biologics that target some of this already. We have, tezepelumab, an anti-TSLP. We have an anti-IL-4/13 dupilumab. And you see that many of these overlapped, but we do have significantly more genetic variants associated with childhood onset asthma than genetic variants associated with just adult onset asthma. And I think that's important as we think about in what patients would genetics or genomics be helpful in predicting response. We also know that asthma-related phenotypes and traits are heritable, like we know that IgE has associations with, for instance, the HLO region. We know the lung function also could have has some genetic components to it. So I would say that maybe the closest to translation now is the IL-4 receptor alpha variant in dupilumab response. And maybe I'm being hopeful, but I think it's probably one of the furthest along. And so this variant in the IL-4 receptor alpha chain is a single nucleotide polymorphism resulting in a glutamine to arginine. So the Q is the wild type and the mutant is the arginine, the R. So arginine substitution at position 576. And this has been shown to be associated with asthma exacerbations, worse or more severe atopic dermatitis, and increased IgE response. In this study, which I'll highlight again on the next slide, would see that it also promoted mice who had this variant were more likely to have conversion of induced Treg cells towards a T helper Th17 cell fate, driving a Th2, Th17 inflammation, which we know is a more severe asthma endotype and really manifests as corticosteroid sensitivity and other things. So again, in this study, I believe this is from Dr. Talal Chatila's lab at Boston Children's and in collaboration with Wonderful Botanical, they showed that this variant enhances lung inflammation. And so they used wild-type mice and these IL-4RA knock-in mice who had this variant, and then they challenged them with house dust mite. And what they found is that the mice carrying the variant, they demonstrated higher levels of lung inflammation. And you see the inflammation score, it talks about, how many cells, and you can see that was higher in the mice carrying the variant post-challenge. We also see that the PAS score, which tells us what the airway epithelium is doing in response to this challenge, was also higher in patients in these mice with this variant. They also found that these mice had higher reactivity. So you see bottom here, the methacholine response was higher in mice carrying this variant. And there were higher absolute numbers of tissue eosinophils, neutrophils, and lymphocytes. And here I'm just showing the eosinophils and neutrophils. So this polymorphism enhances lung inflammation. In other more translational studies, now looking at human studies, Dr. Peggy Lai and Dr. Botanical in this study showed that this polymorphism modifies the impact of asthma triggers and pollutants. So they found a gene by environment interaction between this variant and high endotoxin exposure, where if you add the wild type, the QQ, you would see that it was almost a protective effect as these children in the school inner city asthma study, CCAS study, were exposed to endotoxins. And would see that if you were either heterozygous or homozygous for these polymorphism, you had a higher risk. You're more likely to experience asthma symptoms based on as you're exposed to endotoxins. Again, suggesting that this has translational potential. But we're not there yet. So this is an ongoing study by Dr. Botanical and group, and it's the IDEA study, and this is a genotype stratified phase 4 placebo-controlled randomized study. And so, patients have been randomized based on if they have the wild type allele, they're heterozygous for the mutant allele, or they're homozygous, and they've been given dupilumab placebo. And part of the goals, I believe, is also to look at response, how this impacts response. So maybe we'll get there someday. It's one of the closer ones to translation. And then moving on to other approaches that we've used to try and identify biomarkers, we've looked at genetic risk scores. And genetic risk scores offer promise in identifying response biomarkers. First of all, one of the most appealing parts of genetic risk scores is that they're often more readily available in large biobanks like the UK Biobank. You don't have to go do an extra assay, as we often do with proteomics or metabolomic studies. Genes are further upstream, and they influence protein or metabolite levels, and they're more stable than the downstream omics, like protein and metabolite levels, that can be susceptible to the environment and to other factors, like clinical status or disease severity, asthma medications, the environment, or diet. So in this study, we had sought to see if we could identify genetic risk scores predictive of response to the anti-omalizumab, anti-IL-5, anti-IL-4/13 dupilumab. And this was work done in collaboration with Dr. Matthew Moll's team at the Channing. And we defined exacerbations as a greater than 50% reduction in exacerbations over 12 months of therapy. So as I've already shared the limitation of that approach. And we looked at genetic risk scores associated with Th1, Th2, or Th17 variants, and also added IL-4, some IL-4 receptor variants that were available. And in our final analysis, we had 22 genetic risk scores and 14 single nucleotide polymorphisms. We tried to validate our study in an external cohort using the All of Us research program. What we found was that the IL-21 related variants were associated with omalizumab response. So you see here, the higher the better. It was higher in responders. And this replicated actually in the all of us quarter, even though you see the lower threshold here crossed the null value, which wasn't surprising since that was the discovery cohort, the validation cohort. For dupilumab, it was a more interesting story where we saw the same direction with the omalizumab response in the M.Ch.B biobank. However, when we looked in all of us, it was flipped. It was one of the top variants associated with dupilumab response, also in the all of us cohort. However, it was in the opposite direction. And this, for us, suggested cohort heterogeneity. The all of us cohort was just more severe. And possible some other pathway-specific effects that we weren't able to evaluate in this study. So briefly to talk about IL-21, and maybe this could potentially in the near future be a unified biomarker for omalizumab and dupilumab response. So this is a study, actually a very nice study. They initially used a murine model, and they showed that IL-21 modified allergic inflammation. But before I get to this, is to say that a part of the paper I'm not showing is that IL-9, they found IL-9 was important for sensitization and acute inflammation and early exposure. Then they looked at a chronic inflammatory model where the mice were, these B6 mice were also exposed to house dust mite. They were re-exposed or re-challenged with house dust mite. What they found is that almost when these mice are challenged and you look at IL-9 and IL-21 levels in their bronchoalveolar lavage fluid, you do see significantly elevated levels of IL-9 and IL-21. If you looked at the cells, you see there was an increase in the total number of cells. However, if you gave anti-IL-21 to this chronic inflammatory model, it blocked that increase. Again, suggesting that this is probably IL-21 driving this. Either you give anti-IL-21 antibody alone, or you give it in conjunction with anti-IL-9. You see that if you give just the anti-IL-9, that does not significantly change the levels of these cells. So in these mice, you would see there is elevated neutrophils and eosinophils, and they concluded that IL-21 and IL-9 really synergistically boost Th2 inflammation and ILC2 following allergen challenge. What was great about this study was that then they moved forward in that same study and they tried to include a translational component. And so they got some patients with who had allergies and some allergic asthmatics. So the allergic controls do not have asthma, and then the allergic asthmatics. And they did looked at their BAL fluid and did ELISA. And what they found that I'm showing here from their graphical abstract is that IL-9 and IL-21 levels were elevated. And then they did single-cell RNA-seq on endobronchial brushings, and they were able to show that there was increased IL-21 receptor transcripts and predicted IL-21 ligand activity. And also suggesting that even though this was initially in mice and they saw this possible has possibly has a translational potential. I think so that's a nice segue, I think, to talk about transcriptomics. So we've talked about asthma being a complex chronic inflammatory disease. What's important to note is that the clinical phenotype does not perfectly correlate with endotypes, and often the biomarkers we're using are imperfect. For instance, the peripheral blood eosinophil count is an imperfect proxy for airway eosinophilia. So transcriptomics-based endotyping has been helpful in improving really our mechanistic understanding of asthma and in probing treatment response. The transcriptomic endotypes often correlate better with what's going on at the cellular level in the airways than just our peripheral biomarkers. And so it allows mechanistic probing and pathway level resolution, and it can provide better resolution in identifying potential responders. So in this small study, they looked at 8 patients, 4 who got mepolizumab or reslizumab, both anti-IL-5s, or who got anti-IL-4, IL-13, dupilumab. One of the things, and then they looked at what it did to their cells at 0 months, at baseline, 1 month of therapy, and then 6 months of therapy. They did single-cell RNA-seq. What they found was that the proportions of T cells, natural killer cells, myeloid cells, and B cells, as shown in B, did not change. However, and I don't know how if you guys can see this, however, the composition of the classical monocytes changed, where you would see that irrespective of the biologic that the patient was getting, the IL-1 beta positive classical monocytes were reduced. You can see 016. And this is irrespective of what they were getting was an anti-IL-4, receptor alpha, and anti-IL-5. And we see that the S100A positive classical monocytes went up in these patients. But if you look at this heat map, what was important is that the transcriptional landscape also changed. At 1 month of treatment, if you look at whether they were getting mepolizumab or reslizumab, anti-IL-5, anti-IL-4/13, you see that 1 month, things had not really changed. However, at 6 months, you can see it's a lot of pinks. There had been suppression of or reduced expression of a lot of genes that had to do with NF-kappa B signaling and so they concluded that these biologics lead to global suppression of T2 signatures and particularly a downregulation of the NF-kappa B pathway, and this is irrespective of the biologic. And again, suggesting that transcriptomic changes or transcriptomics might be helpful in predicting response. So one question you might ask is, these changes we've said, like I just presented, is after treatment. So, the patient had to be after. No, we need things that start and help us to guide selection. But what I would say here is that while identifying biomarkers to guide selection before initiation would be best, early response biomarkers might still be helpful because they could help us in knowing if to switch now or later. So in this study, we looked at patients who got mepolizumab, omalizumab, or dupilumab using, again, the Mass General Brigham RPDR or AHR system. We looked at patients whose lung function had improved on these biologics. So either the slope improved, they were declining, or that was reversed when they started biologics, or they actually had an upward trend where their lung function improved. So these are all responders. We looked at the time to the maximal on-treatment lung function. And what you see for omalizumab, that was almost a year, 48 weeks. For mepolizumab, rather, that was almost a year. For omalizumab, it was about 6 months. For dupilumab, it was about 1 year, 15 months. And we do see that there are some patients whose maximum on-treatment lung function, if they were still coming to our MGB, really did not occur till 2 years or almost 3 years down the line. Again, suggesting to us that it would be helpful if we could find something that would tell us this person at 16 weeks, for instance, this person is likely to be a responder at 1 year. It's still a valuable biomarker. So to highlight this, this study, MB.Ch.B study, tried to do that. The nasal transcriptome changes differently based on anti-IL-5 therapy or response. And this was a double-blind randomized trial of children. They were from low-income urban centers. This patient had nasal lavage. Transcriptome also had viral panels to know if exacerbations were virally induced or non-virally induced. And there was evaluation of gene modules. What I'll first note is that even though they studied, they tried their best to look at not just one response domain. They looked at exacerbations, they looked at, patient-reported outcomes, they used the CASI, and they looked at lung function. None of these other outcomes improved, again, highlighting the fact that biomarkers could be response domain specific. Only exacerbation improved. And as shown here, there was like a 27% reduction in patients in the mepolizumab group. There wasn't, than the placebo group. We do see that when you look at time to exacerbations, though, that was also not significant. However, when they looked at nasal gene expression modules, they found that those were associated with exacerbation response. So, for instance, and just to orient you a little to the 12 things that they found were significantly associated, modules that were significantly associated with mepolizumab response is what is on this figure. The greens are the mepolizumab-treated patients. The red are the placebo-treated patients. If it's hard for you to, which I found sometimes hard to comprehend, this column will show you what happened, where you would see that the eosinophil signatures were reduced in the mepolizumab group and placebo group not significant, as expected, is anti-IL-5s. And we do see part of that is icosanoid metabolism, which is important because we'll talk about metabolomics soon. And you can see other things that went up, but other things that they highlighted was extracellular matrix production was higher in the mepolizumab treated group, but it was definitely higher in patients who continue to exacerbate. And then the TGF-beta, which we know is important in airway remodeling and fibrotic changes in the lung, was also higher in the mepolizumab group, but not significant in placebo. And so, that again suggests that, yes, transcriptomics could offer a way of us studying response, what is different between patients who responded or not, and then use that to guide future therapy. So moving on to proteomics, we know that, cytokines and chemokines are proteins, and biologics commonly target cytokines or their receptors. So it makes sense that it's possible that these proteins being mutable compared to, say, genes, may reflect changes induced by biologic therapy. And now we also know that we also have multiple protein assays available. We have SomaScan or Link, among others. So in this study, they also looked at mepolizumab, and they tried to compare features of patients who responded to patients who did not respond. What they found is that patients who did not respond had high sputum protein expression of neutrophil activation and degranulation markers. They had high alarmin, suggesting some form of epithelial damage, IL-33, TSLP, GM-CSF. They also had elevated pro-inflammatory mediators, like IL-6, IL-1 beta. And patients who had this, they did not have improvement in FEV1. They continued to exacerbate even on mepolizumab. In other work that we've done, we looked at SomaScan, SomaLogic, and looked again at response. This time it was on a count scale, so a negative binomial scale, to try and identify biomarkers of response to omalizumab and mepolizumab. We did find a number of factors or proteins that were associated with response to omalizumab, much more than we did for mepolizumab. For omalizumab, for instance, we found LTB4 receptor was associated with poor response, and LTB4 receptor is receptor for leukotriene B4, which drives neutrophil recruitment and inflammation, so that's not surprising. We had mucin 1 was one of the things that was actually associated with omalizumab response. And then for mepolizumab, we had GDF8 or myostatin and lysyl oxidase. when we did a protein-protein interaction network to see what proteins or what pathways might be associated, one of the things in gene set enrichment analysis, we also found strong associations with extracellular matrix organization and extracellular structure organization. So similar to some of the things that they found in the MOPIT study. And these 5 clusters had the strongest associations with response. And we see again that the TGF signaling pathway came up as one of the things associated with response. And so I'll end by talking about metabolomics. Metabolites are reflective of multiple intrinsic and extrinsic factors. So, if we have these are small molecules produced or modified by metabolic processes, and they could be lipids, amino acids, or sugars. And then metabolomics is the large-scale study of these small molecules and biological samples. And often they reflect real-time physiological disease states. So they're more susceptible to a patient's disease state, to medications, to the environment, to diet. So we do know from much of the work actually heralded by Dr. Jessica Lascaux at the Channing, we know that metabolites correlate with multiple asthma traits and phenotypes. So they may correlate with exacerbation burden, with asthma severity, with lung function, with ongoing asthma treatment, are you using your medication or not, and what's the burden, how much corticosteroids have you used recently, and also to response to corticosteroids. And importantly, this Nature Med paper also showed that metabolomics could also be used to really probe side effects to corticosteroids, in this case, adrenal suppression. So one of the recent papers led by Yulu Chen, also in Dr. Lascaux' lab, we found the metabolites can predict asthma exacerbations. And here, the takeaway here is that if you use our clinical biomarkers, the things we think would predict response or would predict exacerbations, a lot of these were not significantly different from a coin toss. However, once you add in metabolites, or the ratios, you see the significant improvement in our predictive accuracy. Again, suggesting the metabolomics does have potential to get us where we want to go, identifying biomarkers. So one of the first studies to do that is the SOMOSA study. This was a prospective cohort of over 200 severe asthma patients. And these patients had to have at least 2 exacerbations at baseline. So these were, highly exacerbating patients. And we saw that in those patients, leukotriene E4 was actually higher in patients who responded. And you can see baseline and week 16, baseline week 16. And you see the 2,3-dinyl 11 beta PGF2 alpha which is a urinary biomarker, urinary marker of PGD2, was also significantly different from baseline and week 16, regardless of if you were a responder or non-responder. But when they split it, it was only the leukotriene E4 that was associated with this global effectiveness treatment measure, which is like a subjective way a doctor assesses if they think you've improved on the bio or how you're doing in terms of symptoms, lung function, exacerbations, medication use, quality of life. However, one of the, I think, top takeaways from that study is that breath or volatile organic compounds, so breath measured in the breath, and plasma lipids were associated with response. And you can see here some of those I've listed here in this table, but you can see the area under the receiver operating characteristic curve were really high when they used breath metabolites or some of the plasma lipids, notably ceramides and sphingomyelins, which replicates some of the work that Dr. Lascaux' team has been doing. So what we're doing currently is leveraging this cohort, this prospective cohort, and seeing if we can actually identify biomarkers to omalizumab, which was the treatment these patients went on in this prospective cohort. And I have to shout out to Andrea Aparicio and Talgo, who have basically led almost all the analysis, and taken the lead on this project. And so we're hoping we'll get something from there. But what I can tell you now that we already have is that this metabolite-based framework could accurately predict oral corticosteroid response, so a reduction of 50% or more of OCS dose, particularly from week 16. And so what does that mean? At baseline, if we use this metabolite framework, we see that the AUC is not as impressive at week 4. However, even if we use that and we started a patient on omalizumab based on clinical judgment, at week 16, we can know, with an accuracy of 0.8, if this patient is likely to be a responder, meaning if they're on oral corticosteroids, if their dose will be halved or eliminated, or if they would at least be considered a responder in terms of oral corticosteroid use by a reduction of 50% or more at 52 weeks. So going back to this response, you start first and then we go. And so just to put this all together, we know that there's a need for more accurate biomarkers to guide biologic selection. And, I talked initially about the validated biomarkers. We have the peripheral blood eosinophil count, which again is maybe the most used biomarker in biologic selection. We often see 150 cells per microliter being used as the cutoff, but we know that it's better if it's actually greater than 300 cells per microliter for a lot of the anti-IL-5s and the anti-IL-4/13. And then we do know that this is associated with great response to at least 6 of the 7 approved biologics, and possibly even to omalizumab from some of the earlier studies. For the exhaled nitric oxide, this predicts response to tezepelumab and dupilumab and somewhat to omalizumab. So a useful biomarker in patients who will be eligible for those therapies, but not to the anti-IL-5s in general. So we know asthma is a network disease, and I question that I think 4 or 5 years ago now, Dr. Ed Silverman, as Dr. Scott Wise was encouraging me to expand my research program to include prediction. And I remember meeting with him, and I was saying, okay, this is the omics I'm going to do. And he asked me a question that I still ponder on till today. And it's, okay, tell me the omics, what do you think the omics category that might find these biomarkers to these biologics would be? What should I be doing? Genomics? Should we be doing transcriptomics? Should we be doing? And you might say we should do everything, maybe. But sometimes resources are not for everything. But we know that from the prior studies, it does seem that the genetics or genomics might play a bigger role in childhood onset asthma, particularly in allergic asthma versus like maybe eosinophilic or non-T2 asthma. We know that transcriptomics are very helpful in unraveling cellular mechanisms and might be particularly helpful if we want to think about pathways that are related to different response domains. Proteins and metabolites being more mutable may reflect changes induced by these cytokine-targeted biologic therapies. But as I've highlighted in the SOMOSA study, which I just realized, one of the points, which I don't know if you saw here, is that even though we had this, what I'll consider great AUC for predicting 50% reduction in extubations by 52 weeks, there was no association with extubation response. Again, suggesting that our predictive accuracy can really depend on, the omics type we're doing and on the response that we selected. So we know that metabolomics may be more helpful, maybe in oral corticosteroid response, even though we know prior studies have also shown some association with extubations. So in the perfect world, if we had all the money, we had all the samples, we could do whatever we wanted to do, a multi-omics approach might provide better and more global resolution, but these need to be usable in the clinic. So whether we're doing proteomics or we're doing metabolomics or epigenomics, if it really can't come back to the clinic, then it's not going to be useful or translatable. So in conclusion, I would say the take home is that for, these biomarkers are close, but clinical translation needs to be prioritized. The best validated predictors today are still clinical and inflammatory. So again, the peripheral blood eosinophil count and the FeNO. I think based on maybe idea and some of the signals that we're seeing, some of the work we're doing, that maybe omalizumab and dupilumab might be where omics may first prove transformational, but there are many other people doing this research across the country, and I'm just excited to see what really gets into the clinic, because a lot of this are just discovery stage. And then the future, we need to integrate omics domains and collect data prospectively and externally validate, and again, make sure that these are possible to be translated to the clinic. And so I'll end with that. I want to say a big thank you to Dr. Scott Wise, who, is fundamental to my being able to expand my research program to include predictors of response, and some of our collaborators, Dr. Lasky Su, Dr. Matt Moore, Tanami Nopsopong, who was my first postdoc, and part of Dr. Lasky Su's team, Andrea, Tao, and Cynthia. And I would like to thank the MGB Severe Asthma Group, particularly Dr. Elliot Israel and Dr. Fanta. Thank you for this opportunity, and also for funding, and a big shout out to American Lung Association, because they were fundamental in me being able to do a lot of this proteomics and metabolomics assays. And I'll take questions. So thank you.
And that was great, Ayobami. Thank you for such an informative and comprehensive view of biomarkers and the possibilities that they may give us insight into which patients might be most responsive, and also insights into possible other interventions. Uh, so there's a question. Can I ask a basic question? The FeNO values is a biomarker for biologic response measured with patients on inhaled steroids, given the potent effect of inhaled steroids in lowering FeNO.
Yes, unfortunately, many of them had measured FeNO in patients who were using biologics or who were using their inhaled steroids. So yes, that's a great limitation, particularly to the observational studies. I think some of the more experimental studies are able to do baseline measurements sometimes of therapy, but most of these were actually measured with patients on inhaled steroids. So great point. I didn't show the slide. Many things do influence FeNO, and inhaled corticosteroids being one of it.
Okay. Well, another question related to the data you showed about extracellular matrix networks. And so, were upregulated ECM networks predictive of a response, or were downregulated ECM networks predictive of a response? What?
So in the Muppets 2 study, actually, it was increased, which makes one think about, is there a restructuring of it was increase in mepolizumab patient, increase in responders, and it makes one wonder, was there a reorganization of what was already ongoing in these patients?
So just to be clear, they were looking at whether there was an increase in ECM expression, extracellular matrix uh, transcriptomics, and found that when there was an increase, that was associated with a response, as opposed to baseline. We're talking about then
Correct.
changes. Is that
Correct.
right?
Correct.
All right. So maybe some repair mechanisms that are actually occurring that are associated with those responses. Okay. Um, and when you showed the metabolomic data and the
Oh,
ability sorry?
sorry, I just saw that there are questions in the chat, but you're good. Yeah.
I don't think there are additional questions in the chat yet. Are you seeing other questions?
Yeah, I see two questions, but you go ahead.
Oh, no,
Go
okay.
ahead.
So then why don't we go ahead with those? Why don't we go ahead with those questions? I'm not seeing them.
Okay, so I have a question here that says, it looks like there are transcripts that go in opposite directions with IL-5 and IL-4 receptor alpha blockade. Was this focusing on the monocyte or macrophage clusters? And that I am not sure. So, for when depending on IL-5 or IL-4 receptor alpha blockade, if they focus specifically on just the monocyte or macrophage clusters, I'm not certain if they did that. And then there's another question from Dr. Barrett. With respect to metabolomics and the variation in metabolites over time, how should studies be controlled to get robust data? How many time points? What exclusions? And by contrast, is there a role for assessment of metabolomics at the time of an exacerbation as opposed to baseline? So great question. I think not just for metabolomics, we're also finding for proteomics, there are a lot of factors that we probably didn't think about before that we have to maybe think about a little more. And I think that the last question about should we be getting metabolomics or even any omics that we're interested in at time of exubations, I think this is very helpful because we might find a lot of times for these patients at baseline, let's say this is their quote unquote steady state, a stable state, we do find that during exubations there's a perturbation. Like a lot of times when people are actually, some studies have shown that if you do look at biomarkers in the blood leading up to a patient having an extubation, you can start seeing upticks of some cytokines that suggest this person is going to have an extubation. And so doing metabolomics at that time will be helpful. I think for the variation in metabolites over time, self-control is very important. But collecting as much as we know about medications, similar to Dr. Fanta's question about the inhaled corticosteroid, what medication that the patient's on, what's their diet? Because again, and that's, I think, the blessing of things like proteomics and metabolomics and being mutable compared to like genomics is also the curse because they can be affected by multiple things. So I think a lot of whether we're getting multiple time points will be helpful, so repeated measures, but really collecting as much as we know about their diet, about their medications, including corticosteroid use and other things that might influence the metabolism are just as helpful.
Mhm. Another question related to the data regarding metabolomics and the ability to taper oral corticosteroids I think it was that in patients who were getting treated with anti-IgE. Um, uh, the study you
Wait, which
showed.
one?
Um,
The one with the AUC? Say
yeah,
that
with
again.
its metabolomics and and the ability to taper oral corticosteroids. Um, was that a placebo-controlled study? And if it wasn't, um, what do we think about, how do you interpret that in relationship to kind of phenocopies for the response? Could the metabolomics be telling us that there are patients who are adrenally suppressed who can't come off, and there are patients who are not adrenally suppressed who can't come off, as opposed to telling us anything about the biology of the disease or predicting really what patients would be able to come off regardless of whether they're being treated, because it's such a
Correct.
high placebo response in that case.
Yeah. And this is not I forgot. This is so we looked at 50% reduction at 52 weeks, right? And this issue you bring up is is an issue that Jessica Lassisu and I and the Somoza group, Ratko Jukanovic and University of Southampton, we talk about every single time. So this wasn't a placebo-controlled study. This was a prospective cohort. So it was about 200
Mhm.
patients repeated at 4 time points. That's the first thing. So that's obviously a challenge and a limitation. However, we did adjust for medication dosing and medication burden. And we think that some of this, also based on a lot of work that Dr. Lascaux has done in the lab, that some of this effects are independent of what steroids people are getting or what dose of ICS they're getting. But yes, there's probably some residual effect based on these medications people are already on.
Mhm. Another question is in the chat is thank you for the very informative presentation. Can you speak a little bit more to how transcriptomics could be used to predict differential response to different dimensions, for example, exacerbations or FEV1? How do you interpret the data in the context of the overlapping underlying pathways?
That's great. I think a lot of it has to do with maybe pathways that might be, let me say, things that might be associated with each response domain. So again, saying the fact that someone's quality of life improved in a therapy did not mean that therapy would be the best in improving extubations or in improving in lung function. So some of this might be just some measures are more subjective than others, one. But the other things are things that are triggering someone's extubations might improve does not necessarily mean, and we see that often in severe asthma clinic, does not mean their lung function has significantly improved. And so I think that as we look at, let's say mucus plugging or airway reactivity may correlate more with lung function, but may not correlate as well with extubation. So some of these are subjectivity, some of these are because different pathways biomarkers may be more sensitive to that response. Not that something different is happening on improving extubations or lung function might also be part of it. And some of this is a statistical artifact that we might not be able to tease apart. Just, extubations is maybe 2 counts per person. Lung function is a continuous scale, so it will have more power.
Okay. Another question relates to the data you showed where you combined, I think, the transcriptomics of all the biologics. And do you think that that's an approach, considering the f fact that there are so many of these are targeting different
No,
pathways?
no, no. Yeah, you're right. So as I presented that slide, I realized, so we did differently.
Oh,
So we did not combine because we would expect, even though we're thinking maybe IO21 might be a unified for allergic inflammation. No, but for that study, the 201 where we replicated in all of us, the prediction models were run for each separate biologic. So sorry, I told you the overall, but if you look at the forest plot, we had
right.
different patients who were on dupilumab and different patients who were on omalizumab. But that's a great, you wouldn't want to combine because they're just eligibility criteria is different and the way we select is just different.
Great. Okay, we've reached the top of the hour. Thank you again. This is a wonderful presentation. Again, I'd encourage you to register for your CME credits and text for that as well. And we look forward to seeing you at the next Asthma Grand Rounds. Thank you again, Ayobami.
Thank you for having me. Bye,
Okay.
everyone.
Bye-bye.